Hi, everybody. If you're just joining now, welcome and thanks for joining us for our October Lunch and Learn, right on the cusp of November. We're glad you could be here today. I'm Julie Brown. I'm the Raptor Migration and Programs Director at the Hawk Migration Association. And I'm running things today with Kat Lauer. She's our Engagement and Communications Coordinator. And we're very excited to welcome Sage Dale, Master's or Raptor Biology student in the Conservation Genetics Lab at Boise State University, who is going to be speaking to us about her research using comparative genomics to characterize immune genes between Holarctic Gyrfalcon and Peregrine Falcon.
I'm so pleased to introduce our speaker today. Sage Dale is a master's raptor biology student in the Conservation Genetics Lab at Boise State University. She's interested in Arctic ecosystem biodiversity and using molecular and computational techniques to enhance traditional ecological methods for studying birds on these landscapes. While at BSU, she had the opportunity to participate in several long-term Gyrfalcon monitoring efforts, including those with the Peregrine Fund in Nome, Alaska, the High Arctic Institute in Northwest Greenland, and with the Natural Science Institute of Iceland. Prior to graduate school, she received her bachelor's in zoology from Oregon State University and has spent several years working various field and research positions throughout the country and abroad. These include working on raptor migration in New Mexico, spotted owl bioacoustics in Oregon, osprey distribution and relatedness studies throughout the southeastern United States, and my personal favorite, kiwi bird conservation in New Zealand. Welcome, Sage, and please feel free to begin your talk when you're ready.
Great. Thank you so much. Thank you all for having me. I'm really excited to share the work of my master's thesis here at Boise State. As you said, I'm part of the Conservation Genetics Lab here, and I'm excited to talk about how we can use comparative genomics as a really powerful tool to explore questions relating to conservation, and in this case, Gyrfalcon and peregrine. So off we go. Oh, okay, sorry, making sure my slides work. Okay, the Arctic is experiencing the most severe climate change on Earth. Several sources support that the Arctic is warming at a rate 3 to 4 times faster than the rest of the planet. One consequence of global and Arctic climate change is novel disease exposure to Arctic communities. This can happen through a few different pathways, including altered wildlife distributions and migration strategies. So as vertebrates move northward, they carry pathogens with them, or directly through the poleward shift of vectors of diseases like biting insects like mosquitoes, flies, and ticks. At greatest risk are immunologically naive or vulnerable species who lack previous experience with new diseases. So pathogens and host immune competence, which is an organism's ability to mount an effective immune response against foreign material, are co-evolutionary forces that have evolved over millions of years. Mechanisms of immune defense are written into species' genomes and contribute towards their adaptive potential during rapid environmental change. Birds are an ideal system for exploring mechanisms of immune competence because they migrate, they have multi-species interactions, they often facilitate disease transmission, and they have a long evolutionary arms race with pathogens. Immunogenomics is a well-studied field for model species like chicken and zebrafinch, but is really limited for Arctic birds. A geographic advantage of nesting at higher latitudes is less frequent exposure to pathogens. As a result, birds nesting in colder and drier climates have less diversity in their immune genes than those living in warmer and wetter conditions. Given accelerating warming throughout Arctic systems, there's a need to identify mechanisms underlying immune competence to monitor populations and assess their vulnerability in a changing Arctic. So the vertebrate immune system consists of the innate and adaptive immune systems. And the innate immune system is preceded only by an organism's physical and chemical barriers. So it acts as a first line of defense against pathogens. It's characterized by its rapid but general response. So in this figure, we can see that antigen X is introduced to the immune system twice over a 28-day period, and both times it responds at the same speed and the same magnitude. The adaptive immune system is a secondary defense and is much more powerful. Here we can generate memory, and it becomes more rapid and efficient with each subsequent pathogen exposure. So here in blue, we can see the adaptive immune response. And when it first encounters antigen X, it has a slower response, but it's more powerful than the innate immune system. And then on its second exposure to the same antigen, it has a much faster and much greater response. So across vertebrate lineages, resistance to pathogens has been primarily attributed to the major histocompatibility complex, which is an important protein in pathogen recognition, primarily in the adaptive immune system. And it's widely accepted that greater genetic diversity in MHC leads to enhanced resistance to pathogens. However, in some avian lineages, there's less variation in this functional gene. Despite having abundant populations worldwide, MHC diversity is very low in falcons, which suggests that they're utilizing a different mechanism of immune defense against novel pathogens. So toll-like receptors are an ancient and fundamental component of the innate immune system. They're located on the inside and outsides of cells that allows them to recognize a wide range of pathogens. They have this extracellular pathogen binding region in blue and an intracellular region on the inside of the cell that communicates with proteins inside and initiates the immunological cascade. Birds are generally thought to have 10 toll-like receptors, and each one has the ability to bind to a different type of pathogen. So toll-like receptors have been well characterized in many model avian species, but research in falcons is limited to lesser kestrel, and there's been no research of toll-like receptors in Arctic avian species. The Gyrfalcon is a specialized Arctic raptor that's shown susceptibility to a suite of avian diseases at lower latitudes. Their breeding distributions are primarily confined to the Arctic, you can see in orange, and their demographics are woven with their prey that mostly consists of other birds. They're therefore able to provide a unique measure of widespread avian health across Arctic ecosystems. Gyrfalcon fall at an important vulnerable intersection. They're Arctic avian species who lack experience with novel pathogens and are therefore immunologically naive. And they're also falcons that we know can't utilize their adaptive immune system in the same way as other vertebrates. In order to understand their mechanisms of immune competence, we need to explore them in context with another falcon who also has distributions throughout the Arctic, but different life history. So this is a good time to review Gyrfalcon evolution. So peregrine and Gyrfalcon diverged 2.77 million years ago. And following that divergence, from peregrine, that proto-hierofalcon experienced a prolonged bottleneck that lasted about 200,000 years. At that point, some of the hierofalcons, including Saker and Lanner falcon, rapidly expanded, but Gyrfalcon remained at a low effective population size until about 10,000 years ago. This reduced population size likely limited expansion and made the effects of natural selection, like their adaptations to cold climate, more prominent as their populations were stabilized for Arctic conditions throughout their evolutionary history. This bottleneck may also indicate a potential founder effect, which is a mechanism that reduces genetic diversity in an isolated population and could have direct implications on their immune competence relative to other closely related falcon species. So today, the peregrine falcon is a globally distributed congener of the gyrfalcon with 19 subspecies and far more lenient ecological requirements. So as global and Arctic climate change continue, Gyrfalcon and peregrine may both be encountering similar threats of novel pathogens, but the genomic mechanisms of immune competence that they are able to utilize might be different. So, given that toll-like receptors have not been explored in either of these species, I first aimed to characterize and describe their evolutionary relationships between Gyrfalcon and peregrine. Here I hypothesized that because of their evolutionary history, Gyrfalcon and peregrine would be sister taxa throughout the toll-like receptor evolutionary history as well. I also hypothesized that their structural diversity of Gyrfalcon would may potentially be less complex than peregrine, and that Gyrfalcon may have less heterozygosity, given that they've likely encountered fewer pathogens throughout their evolutionary history. I also aimed to explore interspecific population diversity, here hypothesizing that because of their life history, Gyrfalcon would have less diversity in their immune genes compared to peregrine. So there's a lot of really cool ways that we can explore these questions, but from a genomics perspective, one of the best ways that we can do this is by utilizing publicly available genomic repositories that store sequencing data for many, many organisms. This basically means that when someone has a genome sequenced, they are able to publish it on one of these websites like NCBI, and then other people can use it. Throughout my talk today, I'll talk a lot about reference genomes and resequenced genomes. Both of these can be found in these genomic repositories. For me, I used NCBI. And a reference genome means a really high quality genome that has really good coverage and depth and strict filtering. And the genes within that genome have been annotated or labeled, so we know which is which. And it can be used as a really good reference or standard when we're comparing other individuals from the same population to that genome. And then a resequenced genome is from a single individual, would also hopefully have good coverage and depth, but we wouldn't expect it to be of the same quality as a reference genome. And then those genes would be unannotated, so it's necessary for us to compare it to a reference genome to be able to find the genes that we're looking for. So for my thesis, I was able to utilize these publicly available resources for both reference and resequenced genomes, which made my project really cost effective because I didn't actually have to go into the field myself to collect the data that I needed to do my thesis, and then go through the process of extracting the DNA and sequencing it. So it's a very cool resource. But that being said, there are many really cool long-term Gyrfalcon monitoring projects going on globally that I would just like to highlight. And these four are the ones that we collaborate with in the Conservation Genetics Lab. And the work that they're doing globally is really valuable for Gyrfalcon conservation. So first we have the Peregrine Fund and Alaska Fish and Game, who have been conducting Gyrfalcon monitoring since the early 2000s with Alaska Fish and Game and the early 2010s with the Peregrine Fund. And they monitor Gyrfalcon and other cliff nesting raptors, in addition to a really wide range of prey species throughout the Seaward Peninsula in Nome, Alaska, which is in northwest Alaska. Then we have the High Arctic Institute, which was founded in the early 2000s, but research there in Northwest Greenland has been going on since the 70s. And they've been conducting Gyrfalcon and peregrine monitoring since that time, in addition to monitoring other avian species like seabirds and passerines. And then finally, we have Ole Nielsen in Iceland, who recently retired from the Natural Science Institute of Iceland. And he's been conducting long-term Gyrfalcon monitoring for over 40 years. So combined, these organizations have decades and decades of information on Gyrfalcon. And this comes in the form of, survey information about productivity, in addition to actual samples collected, either blood or tissue, that have been collected over time. And because of these organizations, we've been able to observe gyrfalcon populations beginning to decline. So in Northwest Greenland, historically occupied gyrfalcon territories have been, for the most part, majorly absent for at least the last 3 years. And in Iceland, gyrfalcon populations experienced a sharp decline following their introduction to highly pathogenic avian influenza, and the populations have certainly not recovered since then. So anyway, I just wanted to highlight the importance of these organizations and these long-term studies that they've been running. Gyrfalcon also nest in really remote areas that can be really, really difficult to access. Most of the times they're not a road, which means that nests are accessed through hiking or ATV or boat. And then once you're at the nest, sometimes they require really large rappels to reach the actual nest from a cliff, and then being able to safely transport chicks up and down the cliffs to obtain samples. So collaborating with organizations that have already really well-established field efforts, like these long-term projects, is completely necessary for collecting new data. But as I also said, they have been collecting their own, samples for many, many years, which, as from a genomics lab, is also really valuable because, hopefully we'll be able to utilize these in years to come. I have been really lucky throughout my thesis. I've been able to visit all 3 of these study sites, even though for my project I wasn't needing to collect new data. I was still able to join them for their field seasons and participate. So I'm very grateful, and they're wonderful organizations and great people working for them. So back to the genomics. Okay, so going about characterizing my toll-like receptor genes for Gyrfalcon and Peregrine, there's a few different ways that I did this. So to begin, I downloaded high-quality reference genomes from NCBI GenBank for Gyrfalcon and Peregrine, and then I also from these toll-like receptors. I searched for unannotated genes using various blast search techniques, and then I performed a synteny analysis and alignment, which I'll go into detail in a second. I also was interested in exploring the actual structural characteristics of the toll-like receptors. So to do this, I used a protein domain mapping tool that classified protein sequences based on conserved motifs or patterns. And for toll-like receptors, those are the pathogen binding regions, which you can see in blue, and the intracellular region in orange. I then compared the motif structure of Gyrfalcon and Peregrine to each other and across the different gene families. Finally, I performed a measure of heterozygosity. So heterozygosity is a common measure used to look at diversity in genetics, and higher heterozygosity indicates greater genetic diversity at a given locus or nucleotide. So to explore heterozygosity, I performed whole genome alignment and variant calling using reference genomes and raw sequencing reads used to generate them. So, oops, sorry. Here we go. So from our phylogenetic tree, we can see that across toll-like receptors for all 5 species, that they group by gene family with strong bootstrap support. Zooming in, we can see that Gyrfalcon and Peregrine are sister taxa to each other, followed by lesser kestrel, followed by zebrafinch, and then chicken. I've chosen toll-like receptor 1b here, but this is consistent for the entire phylogeny of toll-like receptors. This is also what we would expect, given what we already know about the relationship of falcons to each other and to the outgroup species. I also found that for the most part, all 5 species, including our outgroups, had all 10 toll-like receptors, except for a few, which were toll-like receptors 2b, 21, and 2x. There were no annotated genes for toll-like receptor 2b in Gyrfalcon or 21 for Peregrine. And I also found this additional toll-like receptor family that grouped independently from the other toll-like receptors, but was only found in the falcons. It was missing from my outgroup species, and the functional characteristics of that family are unknown. So for the purposes of today, I'll just be talking about 2b and 2x because 21 is still a work in progress. So what I did first to look for my genes was used BLAST searching. So you can think of BLAST as a pattern recognition tool. So you can input a specific sequence and search either a genome or an actual database for that sequence, and it would give you hits if it found a like sequence. So I did this for toll-like receptor 2b and 2x and found no significant hits for either of these genes. Next, I performed a synteny analysis. So synteny means that the order of the genes on the chromosome is the same between species. So for toll-like receptor 2b, that was in peregrine and missing from Gyrfalcon, I chose 20 adjacent genes, 10 upstream and 10 downstream, and then I compared that to the same genomic segment of the Gyrfalcon genome. So as we can see by the order and the connections between the genes, the synteny here is actually very good. The twist that we see is due to an inversion that's happened somewhere along the chromosome, but I wouldn't expect this to have an effect on toll-like receptor 2b, as it's well within the bounds of that inversion. And for the purposes of my alignment, I simply took the reverse complement strand of Gyrfalcon to flip it around and make sure that the genes were lined up in the correct order for Gyrfalcon and Peregrine as I did my alignment. I applied the same methods to look for toll-like receptor 2x, which was present in our falcons, missing from our outgroup. And here, the synteny between peregrine and zebrafinch was fairly good. But again, we can see an inversion that's creating that twist. So for the purposes of my alignment, I took the reverse complement of the zebrafinch genome segment. So when I aligned my genes, or my segments to look for toll-like receptor 2b, I found a 13,000 base pair segment of the Gyrfalcon genome that matched with 99.3% identity to toll-like receptor 2b in peregrine. I input this sequence into BLAST and searched the entire NCBI database and got hits, predicted hits for toll-like receptor 2 for Gyrfalcon and other closely related falcons. Finally, I mapped this gene back to the genomic position where it would fall, and it fell on that synteny block for Gyrfalcon where we would expect it to be. So all of these things support that toll-like receptor 2b is likely present in Gyrfalcon. I applied the same methods to look for toll-like receptor 2x, but this time the region of the zebrafinch genome where peregrine toll-like receptor 2x mapped to had really poor percent identity. So this supports that this gene is likely absent from where we would expect it to be. It's possible it's moved elsewhere, or that it's not there altogether, but further research is needed. So I was also interested in exploring those structural characteristics, and this maybe looks complicated at first, but it's actually pretty simple. So a domain or motif on a protein is basically just a pattern, so it's a specific sequence that characterizes that protein as that protein. So as I said before, for toll-like receptors, that's the pathogen binding region, which is in blue, and that intracellular region that communicates with proteins on the inside of the cell, which is in orange. So here I was able to use an online tool that finds these specific motifs. And by doing this, we can not only confirm that the computational annotations used to annotate the reference genome has done so correctly for our toll-like receptor genes because these motifs are present, but it also allows us to explore how they might vary between species or between gene family. So here we can see that across toll-like receptor families, the domains are mostly conserved. And so in this figure on the right, you see the blue and the orange regions. And those blue is the pathogen binding region, the orange is the intracellular region. And they're in groups of 2. And I realize the text is very small, but the top is the Gyrfalcon and the bottom of the groups of two are peregrine. So we can see that the structure of those is very similar across the protein. We can also see that that intracellular orange region is much more conserved. It's the same length and the same position for almost all of the toll-like receptor families. And then we see in the blue, in those groups of two, there is a little bit more variability, but that is what we would expect, given that that blue region is the pathogen binding region, so it's likely under a little bit more pressure to evolve as it encounters new pathogens. So, yes. Okay, so for heterozygosity, I also found that Gyrfalcon and Peregrine had similar levels of heterozygosity overall, but that the actual distribution of their heterozygous sites was variable across toll-like receptor families. So that's what we can see on the figure on the right. So for example, toll-like receptor 1A for Peregrine in pink had substantially more heterozygous sites than Gyrfalcon, but then toll-like receptor 7 for Gyrfalcon, which is in blue, has much more heterozygosity than Peregrine. We can also explore heterozygosity in context to other species. So I did the same process of variant calling for our 3 outgroup species. And we can see that even though Gyrfalcon and Peregrine, had relatively similar heterozygosity to each other, their heterozygosity is very low compared to these outgroup species. The positions of the heterozygosity or the level of heterozygosity of these species is also what we would expect, but it just shows that, they may be less equipped in their toll-like receptor genes compared to these other species, at least as heterozygosity goes, for encountering new pathogens. So, to summarize all of that, that's a lot of information. I first explored those evolutionary relationships, and I found support that Gyrfalcon and Peregrine were sister taxa to each other throughout the evolutionary history of toll-like receptors. I also found that that toll-like receptor 2B that appeared to be missing in Gyrfalcon was likely there. It was probably just missed in computational annotations due to sequence similarity to some of the other toll-like receptors. And then I also found that additional toll-like receptor 2X family that was in our falcons and missing from our outgroup. Now, is it only in falcons or is it in other species as well? We don't know that, but the important thing is that an additional toll-like receptor family may have important functional implications on the innate immune system as different toll-like receptor families can bind to different types of pathogens. Next, looking at those structural domains, I found that they were mostly conserved across different gene families. So where I thought that Gyrfalcon could potentially have less diversity, they were actually pretty similar to Peregrine. And in general, those intracellular regions were more conserved than those pathogen binding regions, which is what we would expect because the regions on the outside of the cell will be encountering novel pressures more often than the regions inside the cell that are communicating with the same intracellular proteins each time. Finally, exploring heterozygosity. They had similar heterozygosity, but it varied by gene family. And both of those falcon species had substantially less heterozygosity than all 3 of our outgroup species. Now, what selective pressures are actually driving the difference of heterozygosity between Gyrfalcon and Peregrine is something I'm not quite sure of, but I'm excited to look into in the future. And I think what's maybe most interesting is how the heterozygosity varied between them. I think it makes sense that overall Peregrine had more heterozygous sites across their toll-like receptor families. But the fact that toll-like receptor 7 had really high heterozygosity for Gyrfalcon is pretty interesting and kind of suggests that maybe for that specific toll-like receptor, they've been under some type of evolutionary pressure that peregrines haven't experienced. But I don't know. There's many unknowns. Okay, So jumping into the second aim, which was to explore more population diversity interspecifically between Gyrfalcon and Peregrine. To do this, I performed whole genome alignment and variant calling on peregrine genomes and compared these to privately available variant calls for Gyrfalcon. And I had a pretty substantial difference in sample size. So to explore SNP richness, I calculated the average number of single nucleotide polymorphisms per gene for both species. And I did this for the full gene sequence, the coding region, and the binding region. I finally calculated nucleotide diversity, which is a measure of polymorphisms or genetic variation across a sequence or a population. So for those who maybe aren't familiar with a SNP is a single nucleotide polymorphism, and it's a common measure that we use to explore functional genes like toll-like receptors. When we think of a toll-like receptor, we know how important they are for, binding to new proteins. And then when we think of nucleotide sequence, three nucleotides make up an amino acid, and an amino acid is what actually codes for the protein. So a single SNP could alter that three amino acid sequence, and that can actually change the physiochemical properties of the protein, meaning the charge or the hydrophobicity or the polarity. And for a toll-like receptor, that means that it could actually change the way that it's able to interact with a new pathogen. So for our toll-like receptors, I found that across all toll-like receptor families, Gyrfalcon had substantially fewer SNPs than Peregrine. And this is meaningful given that SNPs increase genetic diversity. So greater genetic diversity for toll-like receptors could influence their ability to adapt as they encounter new pathogens. Also, if you remember, different toll-like receptors can bind to different types of pathogens. So here we see that some toll-like receptors have many SNPs, while others have few. And the difference of the number of SNPs between toll-like receptors could contribute towards increased or decreased susceptibility to different types of pathogens. I also was interested in looking at those SNPs within the coding and binding regions for toll-like receptors. So when we think of a gene, we think of it's made up of introns and exons, and the exons are the regions that actually code for the protein. So looking at just those exon regions and then the pathogen binding region that's located within the coding region, is important to actually see like how are those changes affecting the resulting protein. And my results I think are kind of interesting. The majority of toll-like receptor studies on birds have only looked at the pathogen binding region. But the handful that have looked at a full gene and the pathogen binding region found the majority of SNPs were within the pathogen binding region rather than distributed throughout the full gene. And my results are a little bit different. If you look at the y-axis on both of these figures, there's substantially fewer SNPs within the coding region and the pathogen binding region compared to the full gene. So as toll-like receptors go, this is kind of different than what other studies have found. But as genetics and genomics goes, this isn't super surprising, just because mutations within these coding regions are really difficult to maintain. And normally these coding regions are under something called purifying selection. So your gene is actively trying to get rid of mutations that might be harmful to the protein and, mess up the way that it actually functions. So it appears that the falcons, both Gyrfalcon and Peregrine, are probably under stronger purifying selection within their coding regions, whereas other species are under a different type of selection and are experiencing more evolutionary pressure within their pathogen binding region. The last thing that I looked at to explore nucleotide diversity between the two species explore interspecific diversity with nucleotide diversity. And again, this is just another measure of polymorphisms throughout the population. So we can see on the left figure that overall, peregrine had greater nucleotide diversity than Gyrfalcon. And then on the right figure, we can see that that is also the case across all of the toll-like receptor families. And this is just something that I think is really interesting, that uncharacterized toll-like receptor 2X family I included in looking at SNP richness and nucleotide diversity. And it has really high diversity, which I thought was kind of cool just because it's an uncharacterized family. So it could have important, functional implications on the innate immune system. And it could even, be helping compensate for their lack of diversity in some of their adaptive immune genes. So to summarize that second aim, I found support that Gyrfalcon and had less diversity in their toll-like receptor genes compared to peregrine, both in SNP richness and nucleotide diversity. And less diversity in these genes could have important implications and negatively affect their adaptive potential, specifically for the toll-like receptors of Gyrfalcon. So as the Arctic continues to experience rapid climate change and vectors of diseases shift northward, Gyrfalcon remain immunologically naive. Without diversity in their innate and adaptive genes, they may have little adaptive potential and appear to be ill-equipped for novel pathogen exposure. This is really just the beginning. My thesis work has hopefully provided greater fundamental understanding of toll-like receptor characteristics of these two species, and there's a lot of questions that we can begin to ask, including what are the mechanisms driving the difference between Gyrfalcon and peregrine, other than, maybe what we hypothesize. The last thing that I want to say is just that the vertebrate immune system is incredibly complex, and one mechanism does not work alone. Toll-like receptors are one important piece of the puzzle, but to understand the greater picture, we need to look at interactions and mechanisms going on throughout the entire immune system. With that, I would just like to acknowledge everyone that has helped me along the way and made this possible. I have a great committee: Stephanie Gala, Sven Berkey, and Travis Booms. A wonderful lab. Resequenced genomes were provided by Dr. Farouk Al-Aji for Gyrfalcon and published by Jeff Johnson on NCBI for peregrine. And then our wonderful collaborators with the Peregrine Fund, High Arctic Institute, and Natural Science Institute of Iceland. And I think I can take questions. Thank you.
Thank you so much, Sage. It was wonderful to hear all about your research. If anybody has questions for Sage, please feel free to put them in the chat now. We have a few. So I'm going to start with Julie, who says, for evolutionary relationships of toll-like receptors, why were chicken, lesser kestrel, and zebrafinch in particular chosen?
Yeah, that's a great question. So chicken and zebrafinch are both really well characterized model species for many different types of genes, but also for toll-like receptors specifically. We wanted to include chicken because it's probably the best characterized of all, but we also wanted to include a closer relative, and zebrafinch is a really good model wild species. I also included lesser kestrel because throughout my readings, there's a particular author, Al-Qaid, who has done a lot of toll-like receptor work on lesser kestrel. And the reference genome that was available for lesser kestrel was really good. So the toll-like receptors within that genome were really well characterized as well. So it was nice being able to include the closest relative possible for Gyrfalcon and peregrine to see kind of where they fall. And lesser kestrel has also been used when looking at that MHC diversity, which is that other adaptive immune gene. So I thought it would be interesting to include it for comparison.
Amazing. Thank you.
You're welcome.
My question, and I don't know, I'm not a genomic scientist, so forgive me if it doesn't make any sense. But I'm just wondering, you mentioned that Gyrfalcon and Peregrine Falcon, as we know, they're bird hunters. Are they uniquely susceptible to pathogens because they are kind of in this close proximity to other avian species?
Yeah, I think that they, yeah, unfortunately, because they eat so many birds, they experience the effects of diseases like much more than maybe a more well-rounded, predator. I will say Gyrfalcon also eat a lot of ground squirrels. So at least in some places, like in Nome, their diet is ptarmigan and ground squirrels and other birds. But in places like Iceland and Greenland, it's mostly birds. So when like those, though they eat a lot of there's like colony nesting birds, where disease is like rampant potentially. Um, and so yeah, they unfortunately seem to experience it a lot more maybe than another type of predator, but it also, yeah, is a good measure of kind of like how the entire avian ecosystem is maybe doing. If they're not doing well, we can maybe assume that some of those prey species might not be doing so well either.
Right. Yeah,
Yeah.
absolutely. Thank you.
Yeah.
And then Julie would also like to know, do you have plans to continue this research after your thesis and what are your kind of directions for this research?
Yeah, we are going to continue. I'm finishing my master's in the spring, and then I'll be staying on as a PhD student in the conservation genetics lab still, and still getting to work with the Gyrfalcon. My advisor, Stephanie Gala, recently was awarded an NSF grant that will allow us to sequence 100 genomes across Greenland, Iceland, and Alaska. So there's a lot of more questions that we can start to explore intraspecifically across the different populations, to kind of do a deeper dive into the deer falcon and see, what's going on. So yes, I will be here and there's more to come.
That sounds amazing. Congratulations for
Oh,
all of that awesome stuff to come. Gertrude would like to know, she says, I've noticed a reduction in migrant peregrines along the Barrier Beach in New York. We think these are tundra birds. Would they have less resistance to disease than more local introduced peregrines?
that's a really great question. And yes. So yeah, there's, those 19 subspecies of peregrine. And one of the questions I'm hoping to maybe start to, look into even within my thesis, um, is those more northern or long-term migrants, versus, yeah, more local birds that maybe don't move as much. And I haven't quite gotten there, but I think that's like on the right track that the immune competence within subspecies might vary depending on like their migration strategy, um, or yeah, like if they're, an island or mainland resident. There's a great paper by Jeff Johnson who published these Gyrfalcon genomes, and he kind of goes into the subspecies. He goes into it much, much more than I could. And yeah, there's a lot of great ideas, but I think that that is the thought for sure.
Great. And that may kind of lead into JF's question, who says, are we expecting large variations among peregrine subspecies, especially FP tundrius versus other subspecies?
Yeah, great. Led right into it. Very good question. I think that we could see pretty big variation. However, there is, I think, an assumption that intraspecific variation will be less than inter. So even if we do see variation within the peregrine subspecies, um, that variation we would expect it to be less than, gyrfalcon versus peregrine. Um, but I do think that, birds that have spent more time up in the Arctic, could have less immune competence than others, maybe closer at the equator. But that being said, also a lot of the peregrines that hang out up in, the Arctic, like tundrias, they might have longer migration. So they, are bouncing all over the world, versus like a gyrfalcon that's just like staying in the Arctic all the time. So I think that that migration is an important piece, to tie in like where else are they going and are they encountering diseases along the way, even if most of the time they're up in, an area with less diseases.
Absolutely.
Yeah.
Thank you. Gordon would like to know, how could this work help with the conservation of gyrfalcons?
Yeah, that is a great question. I think obviously it can be tricky sometimes to directly tie, yeah, like a descriptive pure science genomics into applied. But I think the important thing is that we're by, characterizing their toll-like receptor genes and just understanding their immune competence better, we can like forecast their resilience a little bit better. We're like a little bit ahead of the curve. We begin to see Gyrfalcon decline, but they're not, their populations haven't completely like wiped out at this point, luckily. But I think, you know, there's many things that can come from this if it's like a management of we identify a Gyrfalcon community or population, say like in Greenland or something that's most at risk. Management strategies would maybe be like, I don't know, work, do something for the prey birds, try to mitigate disease within those, species that they are primarily preying upon. Or like a longer term conservation strategy, which is, way, way far ahead, but like eventually, kind of as they did for the condors with avian influenza, vaccinations could be something to think about like far in the future. But obviously, those things are really far ahead. And unfortunately, this is like this little piece that I've done through my thesis was the first necessary step to hopefully kind of understand how they'll react to diseases to come, without having to like expose them directly. So, yeah.
It's, yeah, it sounds like it's a really important stepping stone to
Yeah.
much bigger goals.
Yeah.
So Jerry would like to know, would your research benefit from comparing Arctic gyrfalcons to falconry gyrfalcons?
Yeah, that's a really cool and interesting idea. I think it could be really interesting. I have thought about this and I wonder how much, like, just because it takes species so long to evolve, like how different birds, like falconry birds or birds that have been in captivity for a long time, would vary from wild birds. But I think that, you could do something there completely of just comparing captive to wild. Um, or, a lot of those captive birds might have, unique like plumage characteristics. So we've talked about how, plumage might also be an indicator of immune competence. So I think, yeah, not only just like out of interest of how do captive birds vary from wild birds, but I also think like the falconry community is just like what an amazing resource of, people and expertise and yeah, more birds that could be maybe used in the future for genomics.
Sorry, we just had a little share screen issue. I don't know if you were able to share your screen again. Either
Okay.
way, it's fine if not.
Okay.
Um, yeah.
Here, I can try.
Yeah, it's fine if you don't. We can just keep it up, or
Okay.
we can just keep it like this if you want. It's
Okay.
fine.
No.
Sorry about
Oh,
that. It
no
must be a
We're
worries.
nearing
setting
the end.
that I
All,
had.
all good.
Okay, and then there's a question from Kyle, and I hope I read this correctly because it's a little long. So Kyle would like to know, makes me wonder if a first step would be to look at genetic movement slash mixing with microsatellites between peregrine falcon populations to see how genetically isolated subspecies are before looking at immunodiversity between populations, so-called subspecies. Is immunodiversity correlated to overall genetic diversity?
That's a good question. I think that the subspecies portion of that has actually been explored really well by that Johnson 2023 paper that I mentioned, where the genomes that I was using for this were actually sequenced and published by him on NCBI. And in that paper, they talk a lot about, yeah, subspecies variation. And he was actually able to use whole genomes rather than just microsatellites. So it's like, across the entire genome of all the genes in the species, the variation has been quantified and described really well by that paper. Um, I think, yeah, I think that immune gene diversity. What was the end of the question? Is immune gene diversity correlated with whole genome diversity?
Yeah, overall diversity, genetic diversity.
That's a good question. Uh I kind of assume maybe it would be, but I'm not exactly sure. I think that's an interesting thought. And hopefully, if we are able to dive into the subspecies genomes a little bit more and yeah, explore within each subspecies their toll-like receptor variation, maybe that could help answer that question. I think maybe toll-like receptor variation could be tied into that, but like I said, also immune system super complex, so I'm sure there's a lot else going on in there.
Absolutely.
Yeah.
Okay, one last question from Yvonne, who says, as a last-ditch effort, if Gyrfalcons were threatened with extinction due to inability to respond to pathogens via their toll-like receptors, could CRISPR be used to introduce protective toll-like receptor genes?
Wow.
I
That's
know.
the idea. I don't know. What an interesting thought though, maybe. Yeah, I mean, ideally that wouldn't happen and I think we're far enough away that, you know, hopefully we can parse out other mechanisms of immune defense, like even if toll-like receptors aren't that missing piece, like their toll-like receptor diversity is less, and we expect that their immune gene diversity in general is less, there could also still be other mechanisms working in the immune system that are helping protect them. But I think that's such an interesting thought, and I think probably before that, maybe like vaccinations would be something that people would try. But that's a very like cool futuristic idea. I don't know the answer, but I like it.
Okay, I think that's the end of the questions portion. I just want to thank everybody. I feel like our group always has really interesting questions. So thanks to everybody for submitting those. And thank you, Sage, for doing such a great job. I'm going to hand it back over to Julie, who's got a few things she wants to share.
Thanks, Kat. Yeah, really great questions. And thank you again, Sage. We really appreciate you taking the time to share your research with us. And it was very fascinating to just take that deep dive into genomics. It's a side of research I rarely get to hear about with raptor research, and that we rarely get to feature. We do a lot of ecology-based stuff on Lunch and Learn. So it was really, really interesting. Um, and for next month, we have another presentation on November 19th, and we're going to be hearing from Bruce Murphy, who's from the Hillerton Banding Station in Ontario, and he's going to be focusing on boreal owl banding and other species that are monitored there. Registration is coming soon for that. And if you'd like programming like Lunch and Learn, we encourage you to join our kettle and become a member. Your support allows us to provide education like Lunch and Learn, like programs like this, and keep Hawk Count up and running, and offer support and grants and things to the Raptor Migration Monitoring Network. So thank you everybody so much for joining, and we hope to see everybody again next month. Thanks again, Sage.
Thank you. Thanks, everyone.
Bye, all.
Bye.
Bye.